Okay, we're live.
Okay, great. So welcome to the Amy seminar series. I'm excited. I'm Peter Fraser. I'm one of the associate directors here in Amy, and we're excited to welcome today Adam Gormley, who's an associate professor of biomedical engineering at Rutgers University, and he's a decorated and very well-funded individual who does just really awesome and interesting work in self-driving labs and their intersection with polymers. Um, Maybe one of the things to highlight about his bio is he's also an entrepreneur. So he's a co-founder of Pleximer Inc., which is leveraging technology that came out of his lab and having a lot of excellent business impact with that. So today he's going to be talking to us about polymer biomaterials in a self-driving lab. So take it away, Adam.
Great. Thank you very much. You see my screen okay? Looks good. Great.
It looks great.
All right. Well, thanks very much. Thanks for the invite. I was really excited when I saw the announcement about the founding of this institute. And so I was delighted when Peter emailed me asking for me to present here because I'm a big fan and I'm definitely looking forward to expanding interactions and collaborating and finding new avenues to work together. So I think this is a great way to get that started. Yeah, so as Peter mentioned, I'm an associate professor in biomedical engineering here at Rutgers. And yeah, as the title says, I'm really excited about self-driving laboratories. The world of self-driving labs has has been the concept has been around for a while, but I would say that the implementation of self-driving labs has really only started to accelerate over the last 5 years. And now there's lots of labs all over the world, really excited just as me about self-driving labs. We all kind of have our own individual niches, our areas of interest. So I'm really interested in self-driving labs for polymer materials and biomaterials and drug delivery. These are the worlds of those three worlds haven't seen that much activity in self-driving labs. And so I'm excited to be one of the people that are really helping to push these efforts and seeing how we can use these platforms for these really important fields. And so the actual process of self-driving labs is pretty much the same for everybody. It's just a closed loop cycle of design, build, test, learn, or however you want to coin those phrases. It's really just a closed loop of really optimization of a problem that you're interested in. So for those of you that aren't very familiar with self-driving labs, so this is an AI institute, so I'm kind of assuming that most people here have some sort of foundational knowledge about machine learning, especially as it applies to material science. So I'm kind of skipping a whole bunch of introduction about that. What may be somewhat new to people who are watching this is the concept of connecting the data science to the physical world in terms of automation and using data science to orchestrate an automated system. And so this is a kind of general overview of how a self-driving lab works. Again, think of this as a closed loop cycle of design, build, test, learn, where each one of these components on the left plays some role in design, build, test, learn. I would say what I would normally start with is just looking down here on the bottom right. If you can see I'm going to quickly just get a little pointer here. The scheduler is really, at the beginning of the day, the most important thing when it comes to actually operating a self-driving lab, right? So it's a set of commands that instructs the instrumentation on what to accomplish, right? So it's going to be usually a sequence of steps: do this, then do that, then do this, then do that. Whether it's pipetting or heating, shaking, or moving samples or measuring samples, right? Ultimately, the scheduler, the orchestrator, is the one that's kind of overseeing the operation. And then there's obviously the controller, the thing that's actually operating the instruments. So whether it's a pipetter or these other, the UV vis, whatever, then the orchestrator sends the commands to the instruments to implement those steps in this kind of orchestrated way. That data then has to be processed, right? So the raw data outputs from some of these instruments are organized in their own unique way. And so that has to be organized, maybe doing some exploratory data analysis on it, and basically putting into a data frame that is useful for a machine learning model and then saved in a very organized fashion. Then we train a machine learning model. Usually it's a model that can predict uncertainties as well as predictions. And so the most common model used in active learning workflows for self-driving labs are Gaussian processes, or GPRs. So that is the most common model because one of the most important things that it does is it outputs the uncertainty of a prediction, which is equally as important from a concept of a self-driving lab as the prediction itself. And so then once you have these predictions and these uncertainties, what you basically do is you create an in silico design space. Basically, okay, here's every possible permutation of the experiment I could possibly do. And what we only have when we've initialized this active learning workflow are a couple seed data points, which is mapping this structure function relationship, this kind of map, this QSAR map, right? And so what the key here is that, okay, if we're trying to model the structure function relationships within the system, and we only have a few ground truth bits of information, what can we do to model the system most effectively, and more importantly, navigate to areas of the design space where we need to learn some more information. And so what you're seeing here is the key concept of active learning, right? So what we're seeing here on the left-hand side, this top left, is imagine this gray line is the structure function model. Obviously, this is a very simple polynomial model, and structure function relationships are really quite complex and multidimensional. But just imagine for the sake of argument that this gray line is what we're trying to understand. This is the material science that we're trying to understand. And these red dots are the ground truth pieces of information that we have based on the first experiment, right? Now, you could draw many different types of lines that are connecting these individual red dots. But what we really want to know is what is the shape of this gray line, which we do not yet know, right? Okay, so that's really the goal of a self-driving lab. So the next thing you're going to do is you're going to train the model and you're going to get predictions and uncertainty, right? So what we're seeing here in these blue shaded areas is basically how much uncertainty we have in our system. In some places, the model has already pretty high certainty about its kind of compositional landscape. Okay, a material with these types of properties already have some performance that we're looking for. And if you're a scientist, what you often don't care about is what you do know. What you usually care about is what you don't know, right? Because it's what we don't know that may provide the most useful information. And so what we actually do then is we use what's called an acquisition function. It doesn't really matter, what that means, but we use an acquisition function to basically estimate what is the next best experiment that we need to do in order to learn the most, right? This is always what we do as physical scientists in the lab. Whenever we do multiple experiments in a closed loop cycle, is we're always trying to do the most productive next experiment to learn the most about our system. This is what we all are always doing. And that's what active learning is doing as well. It's basically trying to sample areas of the parameter space that it doesn't know that much about. And then it basically says, okay, now you have to do these couple experiments in order to learn a lot more, as much as possible. And that basically then sends it to a designer, designs that next experiment, and then again to the scheduler, and then it does those experiments. You have to imagine that this is a closed loop cycle, right? It's continuously sampling the parameter space, updating its predictions, minimizing uncertainty, and trying to understand the structure functional landscape of a material. And at some point, you have to budget, so many cycles of DBTL, maybe some sort of accuracy score, maybe there's a certain material property that we're looking for. At some point, you have to finish the experimentation, call it a day, and then you basically just exploit the model and try to find the materials that you are looking for. And so self-driving labs is just an optimization problem. It's just about doing it as autonomous as possible. That's really about how this works. So everybody in the self-driving lab community has their own kind of flavor of automation. This is my general setup. We love liquid handlers. Most experiments in my life are all about liquid handling. And so a liquid handler for us turns out to be the core set of instrumentation that manages all of this. But then in addition to that, we have all of our other pieces of instrumentation around our robotic arm, such as a custom built light box, which I'll talk about, a UV vis, a DLS plate reader, a heater shaker, and even a custom liquid chromatography instrument that we've been building. And so basically what this does is it gives you a fairly reasonable portfolio of instruments in order to be able to do quite a number of diverse experiments. Now, it can't do everything, obviously. There are a lot of experiments that we can't access, but we're always trying to push the envelope of automation so that we can do a wider and wider range of experiments within a closed loop self-driving lab. And so, like I said, this is really our core instrument. There are many different liquid handlers out there. The most common one these days seems to be Opentrons. I think a lot of people have Opentrons instruments. Our instrument is a Hamilton Microlab. We have two of these liquid handlers. We just love it from a hardware perspective because 8 independent channels that can run around the instrument, pick up reagents. It's literally just 8 pipettes on an XYZ stage, and the rest is programming. And in fact, one of the things that we liked about this instrument is that it's very programmable from our perspective. We actually don't use its own software. We wrote all of our own software to do all this. In fact, this is something that we have always emphasized, that if you're interested in the world of self-driving labs, never be beholden to consume to other people's software. It's really best for you to write your own. And so again, our software, which we call Polycraft, operates all of these instrumentations that you're seeing, the liquid handler, the UV-Vis, the DLS, everything like that. And it really gives us a lot of versatility to do a lot of different types of experiments that we're interested in. So I'm going to give you, that's kind of a very quick overview of our hardware and how a self-driving lab works. What I want to do now is give you a brief overview of a couple different example topics, example problems that we're using this system for. This isn't comprehensive. We're trying to apply this platform to lots of different problems, but this is just a snapshot. And what you see here is in the center, we've got this little robot icon. When you see a project which has that icon, that means that the project is fully autonomous, meaning it's completing all cycles of design, build, test, learn fully independently. Just kind of set it and forget it. Whereas there are other projects that do require more human in the loop involvement in order to accomplish them. I really don't believe that we should be automating everything. I think that we should always be practical in what needs to be automated. That includes sample preparation and things like that. And so I'm very practical. I think we should automate what we should be automated, and we should use humans when it's important to. So let's start talking first about polymer chemistry. So actually, I really got into all this, like most people in this community, got into this kind of by mistake. When I was doing my postdoc at Imperial College London in 2014 to 2016, I was working on a project. Actually, it was a nanoparticle-based biosensing project, and we were doing these polymerization reactions. And we saw something happen that should never have happened, which was that we got a polymerization reaction to proceed in an open vessel. If you've ever done polymer chemistry, you know that these reactions are sensitive to air, usually oxygen or water. And when this reaction proceeded in open air, I'm not going to get into the chemistry because it's not important. We were really shocked by that. It was something that was kind of unique. It was the addition of this enzyme that we added, glucose oxidase, and it suddenly allowed us to do a raft polymerization just without sealing it, without degassing it. It was a mistake in our experiment, but it was a serendipitous mistake because when we saw the result, we said, wow, that should not have happened. Why did it happen? And then we chased it, and then sure enough, we found out that our enzyme was degassing the reaction, which was really cool. So we chased that down and we started to say, well, what can we do now with air tolerant polymer chemistry, which was both Cyril Boyer actually discovered the same thing in 2014. We actually published within 1 month of each other, which is pretty interesting, totally independently. And so when we saw Cyril's paper and Cyril saw our paper, we started to collaborate to say, well, what can we do now with air tolerant polymer chemistry? And this is when the three of us, Rob Chapman, Cyril Boyer, and I, figured out that this is really excellent for combinatorial polymer chemistry. Because now we can do polymerization reactions in everyone's favorite labware, which is a well plate. Everyone loves doing all experiments in a well plate, if at all possible. And that was not possible before because they're obviously exposed to air. But now we can actually do really nice combinatorial polymer chemistry with very high control of the reaction in 96 or 384 well plates. So that was the rest of my postdoc. So then when I started my lab here at Rutgers, we were doing the same combinatorial polymer chemistry over and over again. And my students were probably on the verge of carpal tunnel syndrome at this point. And I started to realize that there was a real opportunity to begin automating this because at the end of the day, this is just liquid handling. And there are great instruments out there that are intended to do automated liquid handling, and these are liquid handlers. And so we actually bought our first liquid handler at a laboratory auction back in 2018 and started throughout its software, started programming the whole thing in Python. and started making our own custom polymer synthesizer. And what we landed on was this Polycraft software that I mentioned, which was the ability to do fully automated combinatorial polymer synthesis. And so the basic idea here is we start with an Excel sheet. That Excel sheet, every single row is a new polymer material, and every column is a feature associated with that. So whether it's a homopolymer, a block copolymer, a random heteropolymer, the molecular weight of the polymer, what monomers are you incorporating, and even methods for post-polymerization modifications, all of that is baked into the Excel sheet. And then what we have also is a freezer full of frozen aliquots of all of our reagents. Only one person in our lab is responsible for making these reagents with a very standardized SOP. And when we upload our Excel sheet to our software, it basically says, go to the freezer, pick out these reagents, pop the top, let it thaw, and hit go. And then it'll go all by itself, all the way from liquid preparation to polymerization, and even methods for processing the materials using this automation, which means that all of the work normally associated with making polymer materials has been fully automated for us. In fact, I hired a recent postdoc that's in our lab, and she's the only person in our lab who's been making polymers the old-fashioned way by hand in our lab in several years. We've always been automating all of our chemistry because it's just incredibly convenient, and you don't need to be a polymer chemist now to make polymer materials. Now, one of the things that we were struggling with at the beginning of all this is that we were doing a lot of photochemistry, and a lamp, which most people use, is really not ideally suited, A, for automation, and B, it's not really great for combinatorial chemistry. Because if you look at a lamp, you basically get this kind of radial distribution of light intensity. The center of the wells are going to get more light than the outer ones, which means that the polymers in the center of the wells, in the center of the well plate, are going to convert faster than the ones in the corners, which means that we're really dependent on where the reaction mixture is within the plate itself. And that was really not ideal for us. And so we decided to go ahead and actually create our own custom LED matrix to be able to do combinatorial chemistry in a highly defined manner. So this one is the first example that we built now several years ago. And what we actually have now is this example. So this is each LED is individually addressable, which means that we can control the reaction, the intensity of the light and the time of the light, and on a well-by-well basis. We can control the color of the light, which means that we have quite a lot of control of our polymerization reaction across the entire well plate. It's incredibly powerful. In fact, our first version of the light box, because photochemistry is a very big field, it's not just for polymers. Our first version we actually published open source, and not that many, a few people have recreated it, but I asked my polymer chemist friends, and actually people rather just buy things than solder resistors to a board. And so we're actually now just planning on commercializing this. So we built now Lightbox Pro, which is going to be a much more commercial version that you can just buy relatively inexpensively, so you can do this kind of combinatorial photochemistry pretty easily. And what this has really given us is a much more homogeneous distribution of light. So on the right, what you see is that radial distribution of light intensity, and on the left, you see a very even distribution of light intensity. And this is something that we actually did find to be very important. Now everyone in the lab uses this light box, and we have learned that having even light across every single well in a very defined manner really helps us control our chemistry a lot compared to other methods. Now, Cyril actually continued working on the PETRAF project, and and this is a major aspect of his research, and he published this amazing paper in Agavante Chemi, I think in now 2019, I want to say. And basically, this paper that he published basically said that you can track conversion, the monomer to polymer conversion, by tracking the fluorescence of the photocatalyst. And that was really cool because that meant that rather than having to use NMR, which we typically use to track conversion, you can just use a UV-Vis plate reader to be able to track conversion. And when I saw this paper, I said, my goodness, that is really cool. But you know what's really cool about that? That allows us now to be able to track conversion with our automation. And so that's actually the motivation one of the reasons why we built this multiplex LED array. Because when you look at a combinatorial library of polymers, acrylates will polymerize faster than methacrylates, which means that if you turn on a lamp, that means, and all of them are receiving all the light at the same time, some polymers may be overconverted and some polymers may be underconverted. And again, that's not ideal. What we ideally want is a system whereby each reaction is getting the right amount of reaction light for the right amount of time, the right amount of intensity for the right amount of time. And so this is what we did here is we built this multiplexed array of LEDs. And what you see here on the top left at t equals 0, everywhere you see a color is a polymerization mixture. Not all of the wells have polymers, only where you see color. We then initiate the reaction, and then what's actually happening is the robotic arm is picking up the samples and moving into the UV vis every 10 minutes. It then measures the conversion by fluorescence and then puts it back. Does this every 10 minutes, turns on the light, converts for 10 minutes, puts it on the UV vis, measures how much it converted, back and forth, back and forth. And then what it's doing is because it's tracking conversion over time, as a polymerization reaction goes to completion, it turns the LED off, right? Because we no longer need to convert that reaction anymore. And so what you're seeing here is as time evolves, 0, 30 minutes, 1 hour to 2.5 hours, is the LEDs are slowly turning off because we no longer need to convert those reactions anymore. And so by 2.5 hours, what you see is that all of the polymerization reactions have reached full conversion. Everything is done. No reaction has received more light than it needed. And so this has really been a really fun thing for us because this allows us to really precisely control all of these different reaction mixtures in a very defined way. Now, when we were doing this, all of this that I just showed you was with raft polymerization, which is really one of the most common controlled living radical polymerizations out there. And when we first published our paper doing automated polymerizations, we tried ATRP. Problem is that ATRP is just really air tolerant. It's really sensitive to oxygen, and we couldn't get it to work. And then I was eventually at a polymer chemistry conference, and one of Chris Maniszewski's postdocs came up to me and said, I see that you've never published automated ATRP. I feel like I know why. We solved the problem. You should give it a try. And so he sent us the paper. We gave it a shot. And sure enough, he was absolutely right. We can now finally do ATRP in a well plate automated. The problem that we learned with ATRP is that while it's really great, reliable chemistry, it is more difficult to optimize. Raft is just reliable. It's so easy to do raft polymerizations. ATRP just requires a little bit more optimization in order to find the right ratio of photocatalyst, metal, ligand in order to get a good polymerization reaction. And so it wasn't as straightforward to get ATRP automated as we expected. And so what Cesar actually did in order to be able to optimize this is he just did what we've been doing now for the last several years, which is just train a machine learning model on the chemistry and then use the model to actually predict what are the ideal combination of reagents in order to get the polymerization that you want. With our raft polymerization, our chemistry is determined based on kind of known chemical theory. If you want this molecular weight, change this ratio of monomer to CTA. That is useful for raft, but it is not as useful for ATRP. Instead, what we did is we trained a machine learning model on the ATRP polymerizations. And now what it's doing is it's actually choosing the combination of reagents based on the trained model and not based on the actual chemical theory. And this is actually something that we've been working on now for several years. In fact, a lot of our raft polymerizations do this as well. We are trying to transition from a kind of empirical or chemical theory-based approach for polymerizations to something that is more based on data science, based on seen information. And we call this the Atlas project, which isn't published yet, but the ATRP aspect of it is. And where we think this is all going is the ability to do sequence level control polymerizations, right? Because we have this ability to, with the liquid handler, to be able to add monomers when we want, track conversion, and add things when they've reached a certain amount of conversion. We can kind of tune where monomers exist within a polymer chain as a statistical distribution by tracking their reactivity ratios. But being able to kind of in a sequential manner add monomers as it's converting is something where I think that has not really been fully explored yet, and something that really excites me because as you'll see in a little bit, I am really inspired by proteins, and I just hate that these reactions are statistical. But the question is how close to sequence level control can we get? And I think that this type of platform is how we get as close as possible to sequence control, given the fact that this is still a statistical polymerization. So that's work in progress. Okay, moving on. The next topic that I want to focus on is hydrogels. I mentioned that I'm a biomaterial scientist, drug delivery scientist, and I wanted to see how we can use this platform for a really classic biomaterial drug delivery problem, which is just hydrogel release optimization. So in the world of drug delivery, people are constantly releasing drugs, whether it's small molecules, peptides, or proteins from hydrogels, either synthetic or natural hydrogels. A lot of people do these. I mean, there are probably thousands of people right now who are doing kind of release profile optimization experiments from a hydrogel. And it's extremely common. And the way that you do these types of experiments, which is the good old-fashioned way of doing science, which is to do an experiment at a time and basically track release understand one experiment at a time how changing the polymer composition, the hydrogel composition, influences the release kinetics of the payload that you're trying to release. One of the most classic material systems that people use is alginate, derived from brown seaweed. It's been used in many different FDA-approved products. Relatively safe, all things considered, once you purify out some things. And it's used, like I said, clinically, and it's a it's inexpensive, it's easy to use, and it just cross-links with calcium. It's very, very common hydrogel material. So people have been using alginate to release proteins for a long time. And so we wanted to kind of test the idea that self-driving lab operations and automation and machine learning is going to be useful for optimizing the release profile of a hydrogel system. And so this is our classic design, build, test, learn, closed loop workflow, where we've got basically all these different possible combinations of additives for a hydrogel. We then use the automation to actually prepare these hydrogels and then track the release of our cargo. In this case, what we're going to be doing is we're just going to be tracking the release of fluorescently labeled bovine serum albumin, just a great, kind of garden variety marker for protein release. And then we use active learning to optimize their release profile and then feed that back into the design process and then just close the loop. Now notice that there is a human in the top right corner. This is not a fully autonomous system. And the reason is because time point sampling with a hydrogel is actually a very tricky thing. And it's not something we could figure out how to automate. So the actual process of taking time points is still being done by a human, but everything else is automated within this process. Now, one of the things that we had to overcome as a challenge to make this project possible was viscosity. So liquid handling is really great when your solution is just water or DMSO or methanol. But the moment you've got a viscous sample, like a typical polymer used in hydrogels, it becomes very difficult to reliably aspirate and dispense. So we spent a lot of time figuring out the liquid handling parameters to be able to do automated hydrogel preparation. All of that is heavily documented in the paper. But this is a video of the actual automation going around, obviously, sped up, going around and preparing hundreds of hydrogels, which again would have been very difficult for any human to do to make hundreds of hydrogels. But we managed to make it work. And then we basically did the time point sampling. We ran into some other interesting challenges. I mean, again, this project was new. And as far as we know, no one's ever done active learning on hydrogel release optimization. And so we had to learn a bunch of new things about how to model the system. And one of the things that we weren't sure about was how do you model release? Do you give the raw data points to the system, or do you actually model the line using a classic drug delivery model like the Korsmeyer-Pepys model, which basically models the release kinetics? And we determined that that was actually the right way to go. And so what we're doing is we're actually modeling every release profile and calculating based on the model line this K parameter, which is basically kind of telling you the overall burstiness of the release. Like, is it more zero order, or does it kind of release very quickly at the beginning? And what we're trying to do is minimize K, because the lower the K, the more like linear the release profile, the more metered out the release profile gets. The higher the K, the more you get this kind of like exponential rise at the beginning, and then it plateaus near the end, which is kind of what you're seeing with this gray line. So because we're trying to minimize K. That was the value that we're trying to optimize within our active learning campaign. Basically, how low can we go in terms of K? How zero order of release profile can we get with the system? And what you're seeing are lots of different K values that the system was trying to optimize. And the model did a very good job. Once we figured, what features to engineer, what models to use, obviously there's a lot of work that goes into developing these systems, but ultimately we got an R squared value, like a model prediction accuracy of 0.71, which is pretty good. Like some projects get higher, other projects get lower. 0.71 R squared with a low mean squared error is pretty good. That's what I hope to get out of this. But more importantly, what we really love to see are these kind of explainable AI SHAP plots that you see on the top right here. I don't necessarily want to spend any time going into how to interpret this data because I don't think it's important here. But what's important to note is what these SHAP plots are telling you is how important are individual features within your system. So one of the things that really pops out is time is a really important feature within our system. That is not surprising. Release is dependent on time. So, and I can get into why time is a feature. There's some nuance about that. But the next thing is alginate molecular weight, alginate concentration, cross-linker concentration, cation size, anion valence. This actually all lines up with if you were a good drug delivery scientist who were doing working with alginate, this would actually make total sense. In fact, one of my favorite things was when this paper was in revision, one of the reviewers commented that we didn't learn anything new. He looked at our chatbot and said, all of those things make perfect sense. Why did you have to train a machine learning model in order to tell me something that I already knew? And actually, I was like, thank you very much. You totally just validated the system. You have maybe spent decades learning how alginate hydrogels work. The system figured out in a couple hundred experiments. And so the fact that it aligns with your professional experience means that we've actually done a good job. So thank you. And I think the reviewer was happy with that response. And one of the things that we saw is that the system learned very, very quickly. In fact, this is something that we do see time and time again. We do see gen-on-gen improvements very, very fast. After one seed and 2 generations of active learning, it was tapped out. It already knew the system. We didn't need to do any more design, build, test, learn campaigns. It pretty much mapped out structure, function, behavior of our alginate system, which was great. And so, obviously we did all of this with the model system, bovine serum albumin. We wanted to test this with a really important protein. This is an enzyme we've been working with for a long time now in the lab, called chondroitinase ABC. It's really useful for spinal cord injuries. Again, I don't necessarily need to get into the details of that, but just know that this is a therapeutic protein that we've been struggling to get a zero order release profile for a long time. And because of what we had done with the active learning, we were able to get it pretty much out of the gate. So I think, again, kind of highlighting the value of self-driving labs. This is a very expensive protein. I think it's like $400 for 10 micrograms. If you were to do this release profile optimization the manual old-fashioned way, it would be very expensive. But because one of the things I love to say is that machine learning is a data minimization problem. I think a lot of people think that data science is a big data discipline. In my opinion, active learning is a data minimization problem. It's all about how do you learn the most with the least number of experiments? That's the key to the game of active learning. And that is exactly what we did here, where we basically statistically learned the most while consuming the least amount of protein. That is the biggest value of active learning, in my opinion. In fact, Mike Webb and I are writing a perspective article right now on this, basically talking about how material science may need to transition away from high throughput and actually focus on the lowest throughput possible. Okay, so now I want to talk about protein mimics. I kind of already alluded to this. I am very inspired by proteins. I've been a polymer chemist for a long time, and then when I look at a protein, I'm just like, wow, we've really got nothing in common. And it's really too bad because I love polymers, but proteins are just so spectacular. We are obviously the great products of really beautiful, well-folded macromolecules. And so, despite my disappointment about how far we have to go with polymer chemistry to meet protein level order, one of the things I think we have actually learned over the last several years, decades maybe, is that actually this viewpoint that proteins are perfectly ordered macromolecules is actually totally not true. I think we somehow learned this in school that proteins are ordered macromolecules because most of our crystal structures are based on really good, well-ordered proteins. But we have now learned that intrinsically disordered proteins make up about a third of our proteome and play an equally relevant role in our biochemistry as normally well-folded proteins, which actually now gave me a little bit of solace in that, if I think of polymers as usually being very disordered and proteins as being very well ordered, actually is not true. Proteins exist on a spectrum of order to disorder, as do polymers. And so what we've been asking in the lab for many years now is, okay, given the fact that, okay, yes, we are working with statistical polymerizations, how close can we get? How close to ordered proteins can we get? We're never going to be able to get a perfectly ordered globule with the chemistry that we have, but how close can we get? So we've been doing, again, I'm going to be glossing, I'm not going to go into it too much, but we've been spending many years doing closed loop cycles of design, build, test, learn, building polymer libraries, characterizing them by dynamic light scattering and mainly small angle X-ray scattering, and then learning based on that information, and then updating our machine learning models to be able to basically navigate a really complex structure function relationship in order to be able to define either globular or disordered polymers. Nature has had billions of years to solve this problem, a lot of evolution over a long period of time. We're trying to take a very similar approach of high throughput automation with machine learning to evolve their designs in a very defined way, is really the ethos of this project. And so, although we can't put this system on a beamline at Brookhaven National Lab, we did want to build a self-driving lab using our system. And the obvious analytical instrument to do this was our dynamic light scattering plate reader. It doesn't give you the richness of information that small angle X-ray scattering does, but it does tell you something, and it's automatable. And so we put our DLS plate reader on our automation and basically built out our self-driving lab for single chain polymer nanoparticles. We did a lot of validation of this experiments. Again, I don't necessarily need to get into the details, but what's really important is that the validations worked all very well. The data quality that was coming out after a lot of work was excellent. If you look here on this bottom left-hand corner, this is radius of hydration. So how big is the macromolecule versus molecular weight of the macromolecule. The bigger the molecular weight, the bigger the hydrodynamic radius. And we see that really, really beautifully, meaning that the data quality is very high from our system after we did a lot of work trying to figure out how to manage the data. It's a lot. I can tell you that a lot of work went into this actually, and we get very high predictive accuracies. And so this was very much so a fully autonomous self-driving experiment. We've done this now several times. This is just the most recent one. This ran over last Labor Day weekend all by itself, ran the whole weekend by itself. And basically what we saw, really interestingly, is that it pretty much learned the structure function landscape very quickly. We didn't see that all the time. Most of our other campaigns did worse. This one did it very, very fast. And in fact, what we saw was that, yes, it learned very quickly after the first generation. It actually started to get worse as time went on, which is really fascinating. As one of the things I love about self-driving labs is it's a kind of a Jesus take the wheel experiment, and you're really trying not to manipulate it. And sometimes it does things that are very bizarre, and it's very important that you don't mess with it because otherwise you're just biasing the data. In this case, what it did is because it learned so quickly, it basically didn't know what else to do because we had budgeted 3 rounds of design, build, test, learn, and it started to explore areas of the parameter space that it really didn't know a lot about. It was like, okay, actually, I did a great job in this parameter space, but I don't know anything there. And that's actually why you see the model accuracy going down over time, is because it kept going into areas where it knew less, and that made our models worse. It's really fascinating to watch. We've got a lot of comments from reviewers about that. Our limitations section of this paper is quite long, but I think all of our arguments are valid. And again, we're just being honest in what we publish. And it's just I think these types of anecdotes are just really cool about self-driving labs, and it would be a shame not to publish what looks like negative results are actually kind of interesting results. Okay, so finally, I want to talk about something that's been really important to me. I want to talk about that a lot of people see these types of presentations by myself and others and think I have no ability to do any of this. I get that feedback all the time. My students also tell me this. Because if you think about it, I mean, think of all the skills that you need in order to be able to build a self-driving lab. You need automation, you need machine learning, you need analytical, you need robotics, you need AI, you need the chemistry, the biology, everything, right? That's so much. It's a lot to ask for any one individual. And I think that's why a lot of people are intimidated by this. And so what we've been really working on for the last several years is try to build users guides to bring people along with us. I do have a unique set of skills that I can do this, but it's also important that I also bring others with me and make sure that everybody can work with these tools in an accessible way. So this is our first users guide that we published, a user's guide to machine learning for polymer biomaterials. There's really nothing special about a user's guide for machine learning. There's a lot of them out there. What is, I would say, special maybe about this review, this perspective, is that A is for polymer biomaterials, but most importantly, B is actually this link down here on the bottom right. This link takes you to a Google Colab notebook, which walks you through the syntax of machine learning. I think that's what a lot of the current users guides miss. We all learn through example. We don't need to know what is the definition of a hyperparameter. Like that, you can find that information anywhere. What I think is missing in all of these users guides is the actual syntax. Like, okay, this is how you create a data frame. This is how you do exploratory data analysis. This is how you scale the data. This is how you do a train test split. This is how you fit a model. This is how you do SHAP. This is how you do all this stuff. All of that syntax is there in this Google Colab notebook. You can take a look at it. It's all heavily commented. You can copy this notebook, import your own data. You can learn through experience. And so we're really, I've gotten a lot of great feedback that this has been useful for people, and we're happy to give this to people. But there was always this other animal that was missing, that was just machine learning, but self-driving labs is another beast. And in particular, automation, right? People like myself and others love to show off their complicated, expensive equipment. And that's immediately a put off to a lot of people who would say, I don't have the budget nor the expertise to be able to build such a system. Now, OpenTrons, like I said, has done a really great job at democratizing access to liquid handlers. Many years ago, you could buy an OpenTrons for $5,000. Now it's at least $20,000. So these systems are becoming increasingly less accessible to people. But also what's important is that there are no user's guides to self-driving labs. And so we thought, let's continue our momentum and let's build the first user's guide to a self-driving lab. So this is what we did. It's a whole tutorial. It walks you through what active learning, again, lots of Google Colab notebooks, lots of descriptions, the syntax, everything that walks you through the code associated with active learning and self-driving laboratories, controlling automation, everything you can think of. It's extremely well documented, especially the active learning piece is really great. I'm really proud of it. And so if you're confused about active learning and self-driving labs, I encourage you to check it out. But one of the things that we wanted to do with this tutorial was to show that you don't need a lot of money to do this. I think that scares people away and it's unproductive. And so we wanted to show that building a self-driving liquid handling lab is possible for less than $1,000. So we built two examples of low-cost liquid handlers, or two low-cost self-driving labs. The first one on the left is what we call our pen plotter liquid handler. And then the one on the right is our pipette-driven liquid handler. The one on the right is kind of like a conventional liquid handler. You can build this thing entirely yourself. It's just the XYZ stage. We have the parts list. Everything is heavily documented. Our build guide for building this yourself is 65 pages long with all the CAD drawings. You should be able to build this yourself. But for an even simpler system, the one on the left, we wanted to say, what is the easiest liquid handler we could build for the least amount of money? That's easy for anyone to do. We went to Amazon and basically said, what is the cheapest, easiest, accessible XYZ stage? And that turned out to be a pen plotter. So a pen plotter, as you know, you just screw in a pen and it draws like a little thing. What we did is we just took this pen plotter, which happened to have a good Python API, fortunately, and we basically just changed out the tool, nope, not a pen, but a needle, connected it to a syringe pump, which most labs have. And now we've got a DIY liquid handler that you can buy off Amazon for $500. In fact, what you can see here is then you just put this DIY liquid handler on top of your UV Vis. I don't include the UV Vis in the $1,000 budget because I assume most people have UV Vises if they're doing this. But maybe I'm cheating when I say that. But this is this is what I think of as the minimum example of a self-driving liquid handling lab. I don't think you can get any more simple than this. We also have our pipette-driven one. So in case you're interested in building a slightly more complicated one, but still maintaining a budget, again, the tutorial for building this is all there. That's just an electronic pipette attached to a system that you can easily build yourself. All the code is there. You can do all this yourself. Our GitHub repository for this paper is enormous. I cannot tell you how much time we spent building our GitHub repo for this. It's the most important part of the paper, heavily documented. Every guide, every notebook, everything, parts list, everything is heavily documented. We want people to recreate what we've done and not to feel intimidated by self-driving laboratories. And so we spent I'm very proud of this GitHub repo. So this paper is hopefully going to be published soon in Digital Discovery, and we're hoping that it's going to be useful as an example, as the only, as far as we know, user's guide for self-driving labs. And so finally, we're also getting really excited about robotics. We do a lot of machine vision in the lab. I'm not ready to talk about it yet publicly, but we've been doing a lot of machine vision for lab wear detection, which I'm excited to kind of talk about, hopefully in a few months. So this is a pretty area of interest for us. And we're also really excited about AI agents. We got a heavy push in the lab right now. We think agentic systems are going to be huge for self-driving laboratories. I'm not going to share what we're seeing right now because I'm not ready to talk about it, but yes, it's going to be great. I am really excited about where this is all going. And so I think the next year is going to be really fruitful for self-driving labs and agentic systems. And then finally, we also think that there are opportunities in to commercialize some of these things. This is also my conflict of interest side. Peter already mentioned this. We have a startup company where we're trying to do this. The technology works. The business models are more difficult to figure out. It's really interesting getting into this because you realize how important business models are to startup companies, and so we're learning on the job and growing right now. So we're happy about that. All right, with that, I'll stop here. Thank obviously all my collaborators and students and postdocs for doing all this really great work, my funding agencies, and of course, you for your attention. I'll be happy to take any questions. Thanks very much.
Awesome. Thanks, Adam. That was great. So maybe I'll kick off some questions, and then, folks should feel free to chime in and ask other things that they want to learn about. So one thing I thought was interesting was like you made an offhand comment about